sexual reproduction of bacteria. So sexual reproduction of bacteria or the genetic recombination in bacteria, which is mainly by three ways. One is the transformation, conjugation, and transduction. So this the transformation is by the exchange of genetic material among the bacterial species, and conjugation, which is by the sex pili or the F plus factor, and transduction via a bacteriophage. So these are the three ways of sexual reproduction which can be seen among bacteria. And firstly, we have discussed about transformation. So by transformation, here the genetic recombination takes place. So in nature, there are some bacteria are there after cell death and cell lysis. The genetic material or the genetic constituents which are get released on the environment, and another competent bacteria which take up this genetic constituent. So this is the genetic constituent which are get released into the environment, and this is the recipient cell or the complete bacteria that can encounter this DNA constituents of the bacteria which are released in the environment. So this genetic constituents which are get enters into the recipient cell. So here this is the recipient cell, and here this considered as the donor. So this DNA constituents which are gets enters into the recipient cell. So here this is the donor cell, and here it is a recipient cell, and this in this stage here the constituents or the donor cell constituents enters or take up to the bacteria, take up by bacteria. So consider A, B, C, D, which are the genetic constituents of this donor cell, and here these are the genetic constituents. This is the DNA of the bacteria, and these are the genetic constituents of this bacteria: A, B, capital A, capital B, capital C, and capital D. And by recombination, by recombination here there is a chance to mix up their genetic material. So this is the genetic material which is released in the environment, and a competent bacteria which is able to uptake this bacteria, and thereby it enters into the bacterial cell. And if there is a chance of recombination and a part of this, so there is a, as these two genetic materials which come closer, and there is a chance for recombination. So this there is a chance for recombination. So here there is a chance for recombination. So here there is a chance for recombination. So as a result of recombination, a part or a segment of this donor cells which are get integrated with the bacteria. So here instead of that capital A, there is a mixing of genetic material, or the exchange of genetic material takes place as a result, and these are the uncombined parts left, and here as a result of recombination, a part of this donor cell gets integrated with the bacterial chromosome, and this what we call as the hybrid, and the hybrid quality. So by the mixing up of this genetic material, or by the hybrid, or by recombination, hybrid is formed, and this hybrid characters which can be seen among the descendants, and this hybrid characters which can be seen among the descendants or in the upcoming generation, this hybrid character which can be seen among the upcoming generations. So this is the genetic recombination by means of transformation.